We will cover in this video about the topic that CD8 plus T cells are essential for M1 type macrophages population. So far we have understood that whenever this T cell based therapy is introduced, we have seen the increased population of the immune cells, mainly this M1 type macrophages. So in this video we will understand whether this increase in the population of M1 type macrophages are due to CD8 plus T cells or there is some other reason. So in order to validate that hypothesis, that is CD8 plus T cells recruit the recruit the recruit the M1 type macrophages population at the site of tumors. What it is, they take the immune cells and do the vaccine treatment. One with a depletion of CD8 plus T cells antibody, and one without depletion of CD8 plus T cells antibody. And then when they do this Disney plot, what they have find out is that when the depleting CD8 plus antibodies are not used, they see a increased population of RMC11 and RMC19. In the case of without depleting CD8 plus T cells, and with depleting CD8 plus T cells, we can see that those populations goes down. Whereas the other things that we have seen is that this RMC17 categories are very low in the case where depleting CD8 plus T cell antibodies are not used, but they are very high where depleting CD8 plus T cells antibodies are used. And from the phenotype markers, what they are able to find out is that this RMC7 and RMC11 and RMC19 RMC7 and RMC11 19 types of cells belongs to this M1 type macrophages, whereas RMC17 types of cells or category of cells belongs to M2 type macrophages. And this difference in this uh, upregulation and downregulations are also plotted in the volcano plot, where we can see that there are some other cells categories are also changed, but the highest change has been observed in the RMC19, RMC11 and RMC17. And when they check the CD8 plus T cells in those populations, they can see that the CD8 plus T cells, since in one case CD8 is depleting, so in the case where CD8 plus T cells is depleting, they see a very low signal of the CD45 plus cells, and uh, where the CD8 plus T cells is not depleting, they see a high signal of CD45 plus cells. And these things are true for the RMC11 and 19, since they are overexpressed where CD8 plus T cells are not depleting. And for RMC17, the CD8 plus T cells are depleting, and in that case they see a high population of this RMC17 categories. And these things are reflected also here, that is when they use anti-CD8 plus cells, uh, they see an increased population of CD45 T cells, whereas in the case of only the vaccine where CD8 plus T cells are not depleted, they do not see any CD45 plus cells populations. And the CD45 is kind of a marker of T cells only. So for the RMC17, even though this case 2, that is anti-CD8 plus T cells, has high CD45 plus signals, but they are significantly higher compared to the RMC17 populations, because these M2 type macrophages are very low in that case. Thereby, the CD8 plus CD45 plus cells are very low. And this is the heat map, which further shows that this all the downregulated categories belongs to the macrophages, and also this upregulated downregulated categories belongs to the M1 type macrophages. These are the markers that are upregulated and that are shown on the horizontal axis, upregulated or downregulated. And for this RMC17 and all the upregulated uh, categories of the cells shows the markers for M2 type macrophages. And when they do the this Disney plot of the iNOS cells, here also they can see the clear difference that in the case of vaccine, that is when CD8 plus T cells are not depleting, they see high populations here, but when the CD8 plus T cells are depleting, they see low populations here. And these populations is corresponding to this RMC 1917 or M1 type macrophages. And these things they have validated using the flow cytometry analysis through the manual gating, where they have find out that when they use CD8 plus T cells, anti CD8 plus T cells or CD8 plus T cells is depleting, they see a decrease in the live cells. This However, when they use only the vaccine, they see an increase in the live cells. In the case of RMA Q1, and for this Q1 engraftment mice model, and for this B16F10 engraftment mice models, they see the similar things. That is, when they use anti-CD8 plus T cells, they see a decrease in the live cell populations because tumor cells is accelerating. And when they check the macrophages in this B16F10, they find out that M1 type macrophages goes down when they use CD8 plus T cells, and when they use CD8 plus anti-CD8 plus T cells and anti-CD4 T cells along with the bispecific antibodies. But when they and at the same time they see the increase in M2 type macrophages in that case, compared to the case where they don't use anti-CD8 or anti-CD4 antibodies. Only bispecific antibodies are used to target the tumor cells. So from this it seems like the CD8 plus T cells are essential for the M1 type macrophages recruitment at the site of tumors, because in the case where CD8 plus T cells are not there, you do not see M1 type macrophages populations. So I hope you are convinced or understood how they made this point. If you want to understand more about this experiments, how they did their bioinformatic analysis in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.